G'day, welcome to Navadia. Have you all heard this?
It made me worry that we're rushing the trials in order to rush results for political purposes.
This was developed in such a short amount of time.
Seems like it's rushed, but I know there's a need for it to be rushed. So then you think about, okay, well, what steps or what requirements were taken out in order for them to get this through quickly?
Yeah, we all have. But the real question is, has the COVID vaccine been rushed? In a word, no. And there are multiple reasons why. I'll start with the one that most people generally don't seem to think about. Technology. During the 1990s, a Hungarian-born scientist called Katalin Karikó was studying mRNA in an effort to fight disease. She kept applying for grants and kept getting rejected and kept applying and then kept being rejected. And the reason why this is important is because we need to talk a little bit about mRNA and why it is important. And also, of course, what it does. mRNA is an acronym for messenger ribonucleic acid. It takes the RNA molecule from the cell nucleus where the DNA is stored and transfers it to the cytoplasm, which is where proteins are made. Proteins are basically the building blocks of the cells and have multiple functions as well as structures, signaling, enzymes, and a variety of other things that to do with the cell. Of course, for those really pedantic people out there, yes, sugars, steroids, fats, and other things can also do some of these functions as well, but the bulk of the work tends to fall to proteins. When Karikó was applying for funding grants, she knew that being able to hijack a cell's protein production like a virus could would revolutionize medicine. The potential was not only useful for vaccines, but also the ability to encourage the body to produce enzymes that are missing in some diseases, some growth agents, hormone protections. If it has something to do with proteins, mRNA therapy probably could help. Unfortunately, nobody else was interested. This was because synthetic mRNA was vulnerable to the body's immune system and was difficult to get into the target cells, not to mention the potential side effects and risks from an immune response in some patients. It was deemed risky back then for human trials. In 1990, the University of Wisconsin was able to make mRNA research work in mice. That same year, the Game Boy was released in Europe, Windows 3.0 with DOS compatibility came out for PCs, and Commodore debuted the first 32-bit Amiga. In 2003, the SARS virus, a relative to SARS-CoV-2, which is the virus that causes the disease COVID-19, was first reported in Asia. 8,098 people were infected. 774 people eventually died. Thankfully, this disease did not spread into a worldwide pandemic, which is a pretty big relief given that the fatality rate was 10%. This was the first of two viruses that preceded COVID-19 that scientists began to study for an mRNA vaccine. In 2005, Karikó and her collaborator, Dr. Drew Weissman, was able to figure out how to induce cells to make the foreign mRNA rather than triggering an immune response before it could be delivered into the cell. Unfortunately, this discovery flew under the radar, but for mRNA enthusiasts, this was an absolution as well as the beginning of the race for vaccine development. Initially, vaccine development wasn't on the forefront of everyone's mind in regard to mRNA technology. For example, Derek Rossi, a postdoc fellow in stem cell biology, read the papers published in 2005 and knew that mRNA could be used to sidestep the ethical dilemmas around embryonic stem cell research. Windows XP Professional X64 was released that year, Reddit was launched, Etsy was founded, and the first YouTube video was uploaded. In 2008, a company interested in mRNA technology was founded in Germany by two Turkish immigrants, Ergüz Sayın and Özlem Türeci, whose name I have 100% most likely probably mispronounced. They convinced twin brothers Thomas and Andreas Strungman to invest in a company that would develop vaccines based on mRNA technology. This company's name was BioNTech. Apple iOS App Store was launched, Google released the beta form of Chrome, and Spotify was introduced that same year. In 2009, mRNA was able to successfully reprogram adult cells into pluripotent stem cells, meaning that embryonic stem cells were no longer needed. Rossi informed his colleague Timothy Springer, a biotech entrepreneur. Springer promptly informed a giant in the biotechnology and drug development industry, Robert Langer, at the Massachusetts Institute of Technology. Minecraft was released, and Intel also launched the Core 2 Duo processor i7-500 that year using these specifications because, hey, I might be a nerd, but I know nothing about computers. Chem and microbes are my things. In 2010, Rossi and Springer met Langer to talk about this new technology, more people recruited, and Moderna was founded. Remember, 20 years earlier, Carrico was able to induce mRNA technology to work in mice. In technology terms, in 1990, the world's first internet dial-up access provider came online, the first Adobe Photoshop was released, and IBM released the Power One microprocessor. In comparison, in 2010, StarCraft 2 was released, and Intel launched the Core 2 quad processor Q9500 with these specifications. Again, don't know computers. As you can see, technology has improved somewhat drastically over that 20-year period. We went from DOS-compatible Windows to quad-core processors and Windows 7. In 2012, MERS, another coronavirus, was discovered in Saudi Arabia. This too had a possibility of becoming a worldwide pandemic, but thankfully it didn't because world leaders acted pretty quickly to contain it. Unlike some of the things that we've seen on this go-around, really. Google Play was introduced, Photoshop CS6 came out, and the Raspberry Pi was released in the UK. Raspberries. In 2013, at NIH, it was discovered using a technique in a failed vaccine for a disease called RSV that they were able to genetically modify the virus's proteins to become stable in the body for vaccine development using mRNA. Sony announced the launch of the PS4 that year. Look, I'm not going to bore you with some of the discoveries BioNTech and Moderna had made from when they were founded, but rest assured they had been researching the entire time that they were running. Moderna moved from being a drug company to a vaccine developer due to the problems with the immune responses I mentioned earlier in this video. BioNTech, rather than focusing on getting products out as quickly as possible, they focused more on scientific research. By the time 2013 came around, BioNTech had written 150 papers in 8 years and had 13 compounds in clinical trials. But, like Moderna, was unable to get a product approved. It is worth noting that BioNTech was focusing on vaccine development too. When COVID-19 hit, these two companies had more than 20 years of research combined on mRNA vaccine development. The first rumblings of the potential of mRNA vaccines were around at least 30 years prior to the outbreak. As technology in computers improved, so did the technology for mRNA. BioNTech and Pfizer were working on an mRNA flu vaccine as recently as 2018. Computing power and scientific research goes hand in hand. Do you really think they could sequence DNA using 1990s tech? Today, we have in our hands computers that hold music files larger than some PC games I used to play when I was a kid. In the same vein, do you really think that 30 years of research on mRNA vaccines hadn't sped up? Or science as a whole? We see this all the time. Look how quickly we have gone from finding planets in the early 2000s to today. They took a photo of a black hole, for crying out loud. And yet, for some reason, people think that the vaccine development needs to be slow. I mean, why? We should be expecting to see mRNA vaccines developed at a much faster rate than before because of COVID. Not because they're rushed, but because technology has improved. And that's a good thing. But enough about technology advancements. What about the trials themselves? Weren't they rushed? No. The first reason is this: funding. Getting funding for any scientific research takes time. Lots and lots of time. It's really hard to get. It is competitive, exhausting, and demoralizing. Carrico, the woman who was pushing for mRNA technology in the 1990s, spent the majority of her time being rejected for funding. She even got demoted because she wasn't able to get it. People just didn't care. Very worthy research can take months to even years to be approved. The majority of the time taken up by vaccine research is, in fact, applying for funding. It can take months to years between phases before you can get the funding needed to proceed. Case in point, HIV research took years to be given any funding from Reagan. The first known case in America was in 1981, but Reagan first publicly acknowledged it in 1985. 4 years later. Funding often comes from charities or philanthropists, but the big problem with that one is that causes such as breast cancer can overpower charities for, say, spinal cord injuries or even rare diseases. Charitable funding depends on public's perspective and the motives of the philanthropists. If a multi-billionaire is really interested in space exploration but couldn't give two shits about disease eradication, they're going to put their money into space exploration. And there's nothing wrong with that. I mean, if I was a multi-billionaire, I'd probably give lots of money to microbe research. However, even government funding for public health is extremely competitive, and decisions are influenced by lobbyists and profit-seeking by companies as often as not, as well as the court of public opinion. Australia, for example, was one of the leaders in solar power in the late 90s, but the CSIRO, our National Science and Technology Government Agency, has undergone a series of funding cuts for decades. Because of this, green energy research is now underfunded and we have lost our edge. When the carbon tax was introduced by the Gillard government, it only lasted a few years until Tony Abbott was elected and was scrapped almost immediately. Another example is that carbon capture and storage technology was prioritised over green energy in Australia. These decisions were not made because of a lack of technology, but allowing polluting companies to continue to do so despite what the public wanted. However, with COVID, funding just wasn't a problem. The entire planet was shut down. Nobody could do anything. The entire economy was ground to a halt. Governments needed to get a cure and a vaccine out ASAP. Instead of having to wait years to get a little bit of funding, scientists got it basically the day before they asked for it. And lots of it. Not just that, they were able to overlap the end of the phase of the trials with the beginning of the next one. Usually that's the dotting of the I's and the crossing of the T's. You know, admin stuff. The beginning of the next phase is also admin stuff. There can be months in between trial approvals simply so the folks behind the desk can get through the major stack of applications. With the so-called fast tracking, the application reviews took priority over other non-pandemic applications and thus were reviewed faster. What that doesn't mean is that they were scrutinised less. It just simply means that they were looked at as soon as they came in. They skipped the line, so to say. Thus, they were able to overlap the trials without putting anyone at risk. This overlapping may have shaved off a few weeks or a few months, but when it comes to a rapidly spreading disease that has killed over 3 million people worldwide in a bit over a year, weeks and months matter. Not to mention mutations. The more mutations, the harder it is to develop a vaccine. The longer it takes for a vaccine to be made, the more mutations will occur. Time is of the essence in any infectious disease. If COVID was first reported 6 days earlier and containment efforts were enforced as soon as it was discovered, 3 million deaths could very well be 2 million or under. By the way, I pulled that number out of my ass, but I hope you get my meaning. The fact that scientists were able to produce a vaccine in less than a year because they didn't have to worry about funding kind of blows the mind. Imagine how much better we'd be off if science didn't have to worry about money. Imagine where we'd be if we didn't have to wait 6 months to a year between vaccine trial phases or green technology or any sort of scientific branch. We would be so much better off. Oh, the joys of capitalism. But the other reason why the vaccine was able to be developed so quickly was because of the infectivity of the disease. This is probably the second biggest reason why it was so quickly produced. That and recruiting volunteers was actually pretty easy. A lot of people wanted the vaccine. But before I go on, I'm going to have to touch on the recall of the Johnson&Johnson and AstraZeneca vaccines. This is a video I've definitely been thinking about making. Yes, the Johnson&Johnson and AstraZeneca vaccines have been halted, withdrawn or restricted depending on which country you live in. This is because of a 1 in a million chance of people getting a central venous sinus thrombosis or CVST. This is a blood clot in the brain behind the eyes and you can't prevent it using aspirin. Just in case you didn't know, the safety section of the vaccine trials usually has about 30 to 40,000 people participating, which I will mention a bit later. Unfortunately, those 1 in a million reactions often do not get picked up by trials because there's simply not enough people in it. And even if they were, that singular reaction may still be considered to be within the background rate and not raise any red flags. Sometimes you need 6 or 7 million doses to get the full picture. While that 1 in a million reaction that causes death is an absolute tragedy for the person and their family, it is still significantly better than a 1 in 100 or even a 1 in 50 chance of death and a 100% chance of permanent damage being done to a person's body from COVID. Extreme and rare side effects are unfortunately expected for all vaccines, not just COVID ones. Actually, you can say that for all medical products in general. It is a risk taken in developing any treatment. Some people are allergic to penicillin, but it doesn't make the drug any less life-saving for those that aren't. Risk versus reward is a calculation that always has to be made in these situations. Another thing for you to be aware of is that the AstraZeneca and Johnson&Johnson vaccines are not mRNA vaccines, but instead a viral vector vaccine. These vaccines use a harmless modified adenovirus that delivers the mRNA to your cells, whereas the Pfizer and Moderna vaccines are delivered to your cells via a microlipid. The mRNA vaccines do not have blood clotting associated with them, so the concerns of the anti-vaxxers regarding the mRNA technology do not apply to the recalled vaccines. Every disease has an R0 number. That's the number which describes how contagious a disease is. If a disease has an R0 number of 3, it is expected an infected person would spread it to 3 other people. If a disease has an R0 number of less than 1, the disease is expected to dissipate, while above 1, it is expected to spread. The R0 of measles is 18, which is why we need 98% of people to be vaccinated for herd immunity. Unfortunately, roughly 2% of the population either cannot be vaccinated for measles for medical reasons, or they do not produce antibodies for measles after being vaccinated. This is why measles vaccinations are compulsory if you want to send your kids to a public school in Australia. There's even a no jab, no pay scheme here where people cannot receive government funding for their children if they refuse to get them vaccinated. I have my own personal issues around that, but the scheme worked. Vaccination rates increased after it was introduced. Going back to R0s, for COVID, it is not uniform and it depends on the variant, where the outbreak is occurring, etc., etc. In some areas, the R0 is less than 1, such as in Australia, and in others, it is above 3. But that's not the only thing they need to take into account. It is also the ease of the spread. Let's say that you have a hypothetical disease that has an R0 factor of, let's say, 5. But that disease lasts decades and only spreads through bodily fluids, which is one of the hardest routes for infection. By the way, this hypothetical disease isn't HIV, it is literally purely hypothetical. So if you are infected with this disease and you're expected to spread it to 5 people over decades, that means you might end up infecting someone new every 15 years or so. If you take precautions, you might never spread that disease. Let's go back to vaccine and let's talk about the phases of the vaccine trials because this is important to understand. Basically, phase 1 trials looks to see if it actually works. See if the body contains antigens. There are usually about 20 to 30 people in this trial. Phase 2 helps determine what dose is needed and if it is safe. Usually about 1,000 people are recruited for this phase. Phase 3 is then for effectivity. Phase 3 is then for effectivity of the vaccine as well as to find out if it is safe in larger numbers. Usually around 20 to 40,000 people are needed for this phase. And this is where a vaccine trial might slow down. Science will be able to figure out how effective a vaccine is by how many people within the placebo and drug arms of the trial contract the disease it is supposed to prevent and comparing the numbers, which changes from vaccine to vaccine for a variety of reasons. It might surprise you, but it is considered to be unethical to purposely infect somebody with a potentially deadly disease. It's a bit weird that. Because of that, scientists need to rely on natural spread of the disease amongst the trial cohorts. Basically, they say just go off, do your thing and come back to us if you get sick. Once enough people get sick to infer statistical significance, they can then crunch the numbers to figure out how effective the vaccine is by comparing the number of infections in each arm of the trial. This number will change depending on how many people there are in phase 3 of the trial, how quickly the disease spreads, the results of phase 2, and other variables. For example, if a vaccine needs 100 people to be infected for statistical significance and 95 people from the placebo arm of the trial get sick and 5 people from the drug arm of the trial get sick, Scientists can use this information to determine the estimate of the efficacy of a vaccine. I'm not entirely sure if these numbers mean that this particular hypothetical vaccine is 95% effective. There's a lot of number crunching and stats and mathematics surrounding this part of research, so let's just assume this hypothetical vaccine is not 95% effective. I'm pretty much just giving you random numbers here. Keep in mind, a number of factors can influence the efficacy from one side or the other. COVID in particular can incubate in a person with no symptoms for up to 2 weeks, so statistically there's a chance that folks who've got the vaccine in the third trial might have been infected before they got the vaccination. But this is a big part of why the efficacy portion of the trials needs so many people to be accurate. Let's go back to the previous hypothetical disease. You know, the one that has an R0 of 5 and is in your system for decades. Let's just say for this particular vaccine trial, it needs about 700 people to be infected to figure out how effective this vaccine is. That's going to take a while. Remember, it is expected that an infected person will spread this hypothetical disease to 5 people over decades. On the other hand, in some areas of the world, SARS-CoV-2 has an R0 of about 2 and spreads via aerosols. This means that it might take hours for an infected person to spread it to about 2 people. This is why you wear a mask and socially distance. This high infectivity and fast spread meant that the minimum number of people who needed to get infected with COVID for the statistical significance was going to be infected pretty damn quickly, like in a matter of weeks. This is why one of the reasons why HIV still doesn't have a vaccine, amongst a myriad of other reasons which I won't get into. And this is yet another reason why the COVID vaccine was able to be produced in such a short time. And of course, the final reason why it was produced so quickly is that a lot of vaccine producing companies have had to suspend vaccines for other diseases because COVID took priority. That's why governments were able to get so many shots into so many arms so quickly. So let's sum up. 1. Technology had improved. 2. They had built on prior research. 3. Funding, and lots of it. 4. Fast tracking. 5. The behaviour of the disease. And 6. Prioritise vaccine production for COVID. That's why the COVID vaccine has come out so quickly. Nothing had been skipped, no corners had been cut. All safety trials had undergone the same rigour as any other vaccine trials. It just happened quicker because we needed it to. I hope that helps. And don't forget, scandium, iodine, nitrogen, and cerium. I just want to say thank you to all of my wonderful patrons, especially my $10 Redback Spider patrons, Lauren Hart and Ross Devereux. I definitely want to say a special thank you to my $20 Platypus patron, Paul Butler. But of course, I cannot forget my $50 Inland Taipan patron, Zolfna. Please keep this video in the back of your mind when you hear that the COVID vaccine has been rushed, because it hasn't. Don't forget to share this to all of your anti-vaxxer and COVID denying friends, and I can guarantee you they probably wouldn't listen to me anyway. Happy birthday to you, happy birthday to you, happy birthday to Jazza, happy birthday to you. Yes, I'm sure that brought a tear to your eye.